Okay, good afternoon, everyone, and welcome to our California Rice Virtual Field Day. We thank everyone very much for attending. This is our first virtual field day. As you all know, this is usually held outside at the Rice Experiment Station, so we're trying something a little bit new this year. So we have several speakers. We're starting in about 5 minutes here with our official talks, and we'll have talks about every 15 minutes. In between the talks, we may or may not have time for questions, so please put them into the chat, as Rachel said earlier. We'll go ahead and have a question and answer session at the very end for the last 10 or so minutes of the talks today. So if you can, save your questions for that. As Rachel said before, we are offering Dpr credits. So to get those credits, you need to attend the entire webinar. And then, as she said, there'll be knowledge check polls that'll pop up in between the presentations. And those are for practice. So please, we encourage you to do those polls. And then at the end of the event, there'll be a short quiz that you'll need to pass with a score of 70% or better. And you will also get that exam emailed to you within 24 hours of the webinar. You'll have to complete it within 7 days of receiving it. The CCA credits as well are offered for this course, and there'll be a little QR code at the end. You can just scan that, or you can email the program support unit afterwards. Real quickly before we get started, I just wanted to take a moment to recognize Dr. Kent Mckenzie. Dr. Kent Mckenzie is going to be retiring this year after very many years of service at the Rice Experiment Station and to the rice growers of California. He was a rice breeder at the California Experiment Station since 1988 and the director of the Rice Experiment Station since 2000. Again, he'll be retiring this year, and although we wish we could be there at the experiment station in person, we hope next year we'll be able to be there again. So thank you, Kent. And then just real quickly before we get started, I just want to recognize everyone that's helped make this possible today. So the UCNR Program Support Unit, the California Rice Experiment Station directors and staff, and then the California Rice Research Board and the California Rice Commission, of course, for providing much of the funding. We have industry partners and collaborators that all of us have worked with. And then, of course, our staff and students. And then biggest and most important, the California rice growers themselves. So thank you, everyone. Okay, Bruce, if you want to go ahead and start getting your PowerPoint loaded here. So our next speaker is Dr. Bruce Lindquist. He's a cooperative extension rice specialist at UC Davis in the Department of Plant Sciences. He's going to be speaking to us today about fertility and planting date trials. Go ahead, Bruce.
Hello, everyone. Just a sec here while I get this right. I just want to thank you guys for joining us this afternoon. It's a bit unusual, as Russell was just saying, to do this. However, one thing that it does enable us to do is show a little bit more of what we are doing. We can show some pictures of what's been going on in the season. So it is better in that way. So just try to figure out how to move my screen here. Very sensitive. Okay, so what I'm going to be giving is some of the research updates that we've been working on this year. Certainly not everything, but show some items where there might be something of interest early on. I'm going to talk a little bit about the weather we're having and the smoke. I want to bring up the yield contest, and then hopefully if there's time for questions, we'll be able to discuss those. Having a hard, okay. One of the things that we are looking at is Russell was just talking about lodging. M105 is a really nice variety. It's a little earlier than M206, slightly higher yield potential, but it's been susceptible to lodging, which sort of defeats the purpose in some ways. People want to get it harvested quickly, but if it's lodged, it slows down harvest operations and may impact grain quality. So some growers are actually moving away from M105, and we wanted to do a study this year where we're quantifying the lodging in M105 and to see if there's any way to maybe mitigate against lodging by manipulating nitrogen applications. So we've applied some very high N rates from 120 to 210 pounds pre-plant. And then we have with and without top dress N to see how that may affect lodging as well as yields. Obviously, there's nothing to see yet. You can see the picture in the background there. But one thing we did notice early on, and that might be of interest to some of you, is that we use Serrano in this trial for early season herbicide management. You can clearly see here that M206 is quite a bit more bleached than M105. And you can see that pattern throughout this checkerboard where you got M206 and M105. See a big difference there in the two. Talking with others, Kent, Kasim, they both have sort of seen this before. So if you're using Serrano and M206, it might be something that you're interested in and just kind of keep an eye on. Another thing that we're looking at is one of my students, Carla, is looking at different granular nitrogen sources when we can't use aqua. This year we couldn't, at least for a part of the season, aqua was very limited due to COVID. There was about 10 days where aqua was in very short supply. In previous years, we've had issues with a lot of rain, forcing growers to just flood their fields before they can apply nitrogen. So it's a very relevant question and something we've started to look at. So we have a lot of different treatments. We have ammonium sulfate and urea applied before flooding. We have both of those fertilizers and enhanced efficiency fertilizers applied the same day as planting, so the field is flooded. And we just are applying it when we apply the seed. And then the same group of fertilizers applied 2 weeks after planting. Again, this is the full dose, 2 weeks after planting. And then ammonium sulfate and urea applied 2, 4, 6, and 8 weeks after planting at the ratio shown here at 20, 30, 30, 20%. So most of it coming in the 4 to 6 week range. We're using Super U, Anvol, and coated urea from Agricoat as our enhanced efficiency fertilizers. These slow down the release of fertilizers. So technically you'd be able to apply a lot of it and then it's released slowly. And as it's released, it's being able to be taken up. Part of the problem of applying it all at once is you've got a lot of nitrogen sitting there that's very susceptible to losses, and it would be taken up very inefficiently. So again, we're still early in the season, but Carla's been taking NDVI measurements fairly regularly every week. I took some of her data just from August 6th, which is kind of in that mid to late boot period, and looked at what we're seeing so far. So we have the 0 N here. This is the NDVI off to the left, 0 nitrogen here. And then this grouping is applied before flooding. We have applied right after flooding here, applied 2 weeks after flooding, and then our split applications are here on the far right. And what you can see is that if you can apply it all before flooding, you're getting your highest NDVI. And we've looked at this before with aqua, and typically urea and aqua are very similar in their behavior and use efficiency. So this would be comparable to aqua at the same rate. We're seeing ammonium sulfate at the same rate, giving fairly high NDVI values. If you move across the split applications, again, you're applying it 4 times in this case, we're able to get very similar NDVI values, similar uptake efficiencies, but it's 4 passes across a field. That can get expensive if you're doing it by airplane. So the other options we were looking at is applying it right after flooding, and you can see that tends to be the worst. And then applying it 2 weeks after flooding, which is better than applying it right after flooding, but certainly not as good as some of the others, at least in terms of the early season NDVI values. What we'd hope to see is that some of these enhanced efficiency fertilizers, the Super U Anvil or the AgriCoat, to be a lot better, but they're really, Anvil in this early one seems to be doing a little bit better, but when you go into the 2 weeks after, there's really not a lot of difference between some of these enhanced efficiency fertilizers, which are quite expensive, compared to just applying it all at once. Now, I'd just like to caution that we're looking at NDVI, you know, kind of halfway through the season. We might see very different results at the end. And so hopefully during the winter grower meetings, we can provide you a little bit of an update on that. Another thing that we're looking at that we have been tried to do in the past is looking at just different timing of planting for various varieties. We had hoped to have a mid-May planting date, but we had a lot of rain then, which prevented that date. But we have May 29th, June 12th, and June 26th planting dates out here at the RES. We're looking at M206, M105. Russell just mentioned this new variety, M211, M210, and M209. You can see in this picture taken on August 18th that the flowering heading has occurred in all the varieties except M209 and M211. This week, the June 12th one, we're just starting to see some heading there, and certainly nothing yet in the June 26th. Again, last year, a June 12th planting date was not so uncommon. So that's, some growers are doing that. You really like to be planting in May. So anyways, we're going to be, again, sharing some of this data when we get a little closer to plant, or when we get into the winter grower meetings. So I'd just like to touch base a little bit on some of the temperatures and the smoke that we're seeing. I mean, there's a lot of concern about that, how that might affect rice growth and yields. So I've kind of looked at some of the data, compared it to previous years. And so the first thing that we found in previous studies is you can get sterility in rice when you're getting temperatures above 104 during flowering. Off on this picture on the right, you can see rice at flowering stage. You can see the anthers there. So if you're getting these very high temperatures during that period, it can be a problem. I looked at the 3 CIMA stations that I use, and I'll be showing data from these 3, Davis, Durham, and Williams. So kind of blanketing the larger valley area. And we had anywhere from 2 to 5 days with 100+ temperatures from these stations, and 1 to 2 days where temperatures were above 104. Now those ones that were above 104 were record temperatures, usually around 108, 109. So very high temperatures, but it was only for a couple of days. Certainly, I think the smoke that was very prevalent probably lowered the temperatures, may have been a benefit for those fields where rice was flowering during that really high temperature time. Most of the rice was planted in mid-May, by mid-May this year. And in most cases, rice was past flowering by mid-August when we started to really experience those high temperatures. The other question you might have is, well, what about warm temperatures during grain filling? We've done some analysis on that in previous work, and we've seen no relationship between warmer temperatures during grain fill and yield. It didn't seem to have much of an impact. That said, there could be some issues related to grain quality or disease that might show up. So I just wanted to look in this next slide at these temperatures, looking at historical data. Here we have from 1983 to 2019. This is average August temperatures. I show the high and the low during that time period. And then this is in the middle here, we have the mean of the 3 CIMA stations shown here, with Davis, Williams, and Durham showing the individual ones. And what you can see is historically, an average temperatures of around 95 have been on the high end of temperatures. We're seeing that in this year, and this is through yesterday, so it's as current as I can get it, that the average temperature so far for August is 93.5. So we've seen warmer temperatures historically than this. What's interesting, what is higher is the nighttime temperatures are high. Historically, we've had very few very few Augusts where temperatures have been above 60. This year so far, we're averaging about 62. So quite a bit higher. And so we're probably getting higher nighttime respiration in the plant, which could potentially translate into lower yields. But it also may affect disease. And I'm not sure, Luis may get into that, I'm not sure. Wanted to bring up the smoke as well. 2018, we had some really serious wildfires, particularly up in Redding. And what we saw in that is on these different graphs, Williams, Biggs, and Verona, these are CIMA stations. You can see that there's a real dip in solar radiation. That dip was much greater, closer to the fires. And as you moved away from the fires, like down in Verona, down here near the Sacramento airport, there was still a bit of a dip, but it was much less. In 2018, we did see lower yields. Up in this northwest area, we saw lower yields in the yield contest, and talking to some of the people at the dryers, they were seeing lower yields there. Yields in the rest of the state and the valley were not that impacted, and we saw some pretty good yields. We compare that to this year from 2018. So 2018, the fires started, or the smoke anyways, was serious for about a 3-week period, July 25th to August 15th. This year, the fires actually started on the 15th, and August 16th is when we started to see a real drop in solar radiation. And so we've been about 11 days so far. If you just look at this graph here on the bottom, again, it's the 3 stations that I was talking about. And you can see the solid line across here is data from a normal year, solar radiation. It declines during August because of shortening day length. But if you compare what we're seeing here in this cloud below that, that's due to smoke. We saw some real reductions early on around in August 16th. But overall, there's been a drop of about 30% in solar radiation during the days since the fire started. We've had this really poor air quality. And again, can't say how much that's going to impact yields, but it certainly could, and it may affect other things as well. Certainly, I think that the smoke reduced some of the high temperatures and may have been a benefit in some ways early on. Just want to wrap up with the yield contest. We're going to want to highlight the 2019 winners. We had Greg Driver, Jack Shepard, and Gary Enos getting from anywhere from around 114 to 119 sacks last year. Greg Driver was the lucky one. He won the gator. He won the drawing for that. And we just want to thank our sponsors there listed below that provided funding for that gator. We are planning to run the yield contest again. We've sent out some invitations. Usually on a field day is when we get 90 to 95% of our contestants. We're really low on people that have entered, and we wouldn't be able to really run it with the current amount that we have. So if you're interested, or you're a PCA and you have growers that have some really good looking fields out there, we'd really encourage you to enter the contest. You can enter it by up to this coming Friday, August 28th. We've tried to make it easier. All you need to do is let me know that you're entering. You just need to send me your name, your email, and your phone number to either my email, or you can text me there that you're going to enter, and we can take it up from there. Further details of the contest can be at our website, which is shown down here below if you're not very familiar with that. I don't have time to go into the details of that. With that, I'd like to thank those that are providing funding. Just about everything we've shown you today is funding from you guys through the California Rice Research Board. We really thank you for that. For UC Davis, particularly the Department of Plant Sciences. I've got a great crew of students and technicians that without them wouldn't be able to do any of that. So thank you very much. And I'm not sure if there's any time for questions.
I think there's time for maybe one question, if anyone has one, but so far we don't have anything in the chat. Any questions? So I think I'll keep us moving here. So our next speaker is going to be Dr. Qasim Al-Khatib. He's the CE Rice Specialist at UC Davis in the Department of Plant Sciences. And he's going to be speaking about new herbicides for weed control in California rice. Qasim, go ahead whenever you're ready. I think, is your PowerPoint?
Yes.
Okay.
Just a moment.
Okay.
Good afternoon, and thank you all for coming to this presentation. What I'm going to do today is to talk about the new herbicide that's going to be in the market for California rice in the next season. We've been working with this herbicide for several years, including a large program this year. And before I start, I want to thank the Rice Research Board for supporting our program, and also I want to recognize the graduate student and postdoc who work with me, especially Denise Enns, who is the PhD student working on this project. So the new herbicide, which is going to be in the market next year, is going to be named Loyent. And this is a fluorooxyfen benzyl. That's the common name for this herbicide. Now, this herbicide belongs to synthetic auxin. If you know synthetic auxin, there are several chemotypes within that group, but this is a new chemotype, which is a piclinic acid molecule. If you know oxalic acid, there are 4 or 5 chemotypes. You have the phenoxy herbicide, 2,4-D is an example. You have the benzoic herbicide, dicamba is example. You have the pyridine herbicide, grandistan and aminopyrrolate are example, and and you have the pyrimidine, which is aminocyclopyrrochlor as an example. So this is a new chemotype molecule. Now this herbicide also binds to a receptor in the plant that is new. None of the other auxin type herbicide bind to that receptor, which makes this herbicide unique. It makes it a herbicide that we can use it to control some of the resistant weeds. Loyent is absorbed mostly by foliage, but also absorbed by root. It translocates in the plant by both the phloem and xylem, move to the new growth to meristematic tissue, where you see the death or you see the symptoms from this herbicide. Now, a few things I'm gonna present it here, which gonna be useful if you're gonna use Loyent next year. And I'm summarizing everything we did, the result of everything we did this year, and also the work from the previous year. The rate for this herbicide is gonna be somewhere between 1 to 1/3 pint per acre. I prefer the 1.23. You get better activity from that rate, and also you may delay resistant development when you go with the higher rate. You can apply this herbicide twice. It's pretty safe actually on the crop. And however, I think you're not allowed to exceed more than 2.66 pint per acre, which means 2 applications during the season. We did test many adjuvants to see what's the right adjuvant to use with this herbicide. We tested methylated seed oil, non-ionic surfactant, crop oil concentrate, and also silicone type surfactant. The best result you're going to get is from methylated seed oil. You probably can use non-ionic surfactant and crop oil concentrate, but I will avoid the silicone type surfactant because it seems to reduce the activity of this herbicide. This herbicide gonna be labeled, and I hope that will happen. I haven't seen the label yet, foreground application. I think it's a new herbicide for us. We worry about the drift and what can cause. So I think in the first year or multiple years in the beginning, it's gonna be restricted for ground application. And the volume, probably 10 gallons per acre. You can use more, but I think that's excessive. Probably you will have a good job with 10 gallons per acre. This herbicide is similar to other post-emergence herbicide. We use them in our rice field. You need to lower the water or drain the water to make sure that 70% of the weed is exposed to the herbicide when you apply it. So that's critical. And you can bring the water 24 to 48 hours after that. The herbicide can get in the plant quickly, and within 2 hours, most of the herbicide will be absorbed by the weed. So 24 hours is probably a good time to bring the water after application. The timing of application, you can use it, as I said, it's pretty safe on rice, from 2-leaf stage to 60 days before harvest. However, we did quite a bit of work, and we found the best result we get is 4 to 5 leaf stage. I'm talking here about rice leaf stage. At 2-leaf stage, the rice is too small, the weeds are too small, and if you want to lower the water, if you want to drain the water, some weeds gonna stay under the water level, and the herbicide is not gonna work on them well. So the best result we got is 4 to 5 leaf stage. At later stages, the efficacy declined, and I will get to that. So probably my pick here is stick with 4 to 5 leaf stages. This herbicide is not fantastic by itself. It got to be mixed with something else. It's not the standalone herbicide. So we did a bunch of work to look at what's the mixture and if there is synergistic effect, if there is antagonistic effect. And what we found that we can mix this with a clincher, regiment, granite, grandistant, and we obtain good results. The thing I will avoid the most is to mix it with a propanil. You reduce the activity of this herbicide when you mix it with a propanil. So I would avoid that. And if you want to use a propanil, you got to use them in separate application. We did look at our varieties and how they respond to the herbicide to make sure that we don't have varietal response. And luckily, we did not have any varietal response. All the variety seems tolerant, and it's safe to use this herbicide on all of them. This is a statement about the drift. You know, there is a concern about the drift from this herbicide to nearby crops. And actually, in the South, they had several cases of a drift. So I took this from the label, which you can see, do not apply near beans, soybean, sunflower, tomato, cucurbit, other vegetable, cotton, carrots, grapes, fruit trees, nut trees, corn, grain sorghum, wheat, flour, ornamental or tree, or any other desirable broad plant trees as injury may occur. So this is kind of cover everything. You can use it around anything here, it sounds like. But I will show you some of the work we're doing on the drift to see what's the impact of this herbicide on nearby trees, because I think that's a concern for us. And remember what I said earlier, you cannot use this herbicide by airplane. It's only ground application. Now, one important thing, and I want to stress it, is you cannot use this I'm not recommending this herbicide to use alone. It got to be in a program. It got to be in a program that follow or applied after application of residual herbicide. Otherwise, you're probably going to be disappointed if you apply this herbicide alone. Now, what weed we going to control with this herbicide? That's the main question. I list those, and those based on the work we did in the field. Maybe the label will look different, but we can get control of barnyard grass up to 6 inches tall. Any bigger than that, going to be more difficult. Rice flat sedge, which is usually you don't see it in the field. We see it on the edges of the field. The duck salad, red stem, monocoria, California arrowhead, and water hyssop. We can get control of these. For water grass, we get suppression of the late water grass and early water grass. I think the suppression is more on late water grass and less on early water grass. And it's more of biotype dependent. Now, the label may say you can control small flower sedges and field bulrush, but what we found, it's biotype dependent. Some of the biotype, which we got them from growers, we were able to control them. Some did not show any effect did not get affected by Loyent. And I do have a graduate student, Denise Inns, is working on this to determine what's the base for that. But it's a more of a biotype dependent. Now, this is a picture from study we did in Arbuckle, George Tibbet field, and you can see the control, heavy pressure, sedges, broadleaf weeds, and you can see this is the herbicide at 21 and 60 days, and after treatment, and this is the herbicide applied alone, and the weed control was 50 to 70%. It's not adequate. Another study we did in Willows, this is in Thad Rogers field, and you see the same story, and the weed pressure, this is the control, and the herbicide itself give you 50, 70% in that range, and that's why I stress that you need to mix it with something else. Alone is not going to work. Now, one thing important you notice here, that this Loyent doesn't work on sprinkle top. It's a very weak on sprinkle top. So that's another observation. This is a study we did in pigs. We see a similar thing. And that's what drive me to the next slide, that this herbicide need to be in a program. The picture you see on the left side is Loyent applied after bute, and bute applied at 7.5 pound day of seeding, perfect control, clean field. The same thing can be said about Serrano. We applied after Serrano, we got good yield. Bolero, we also got good control, but not perfect. And this is due to the situation, this field where the weed got ahead of us, and we could not get Bolero until two-leaf stage. At that time, the grass is large and we could not control them. And here is my control treatment. Now, I mentioned about a drift, and we concerned about a drift. So Denise is working on this project to see what's the impact of Loyent on our tree fruits. So he did have a study where new planted pistachio, almond, walnut, prune, and peaches were treated with Loyent. We did the treatment on June 9th. This is kind of coincided with where we expect that Loyent will be applied in the field. And we also have another study where grapes were treated on June 11th with Loyent. Loyent applied at different rate, 0, which is the control, 0.5, 1, 3, and 10% of the use rate. We include methylated seed oil in the spray. Remember one thing, 10% is not a drift rate. Even 3%, I doubt to be a drift rate. But we included in the study, we want to see damage. how the symptoms will look like, and so forth. Thrift rate is going to be 1% to a less than 1%. We measure symptoms. We look at the growth, the leaf size, node, and everything. The only one we take it to yield is the grapes because they are established grapes. Here is the picture for walnut. Not much symptoms we did observe. I will try to, I know I'm getting above my time, but that's because the situation with my presentation. Walnut, not much we did see, very little, and the plant recovered quickly. I think the safest crop we dealt with is walnut. Almond, very little symptoms, and the plant recovered. This photo taking 3 weeks after treatment, you know, just little symptoms, but plant recovered from it. The most sensitive plant to Loyant is pistachio. Extensive damage here, and the plants did not recover from the symptoms. So that's something you guys need to be careful if you use it close to pistachio field. Peaches, we did see some symptoms early on, 3 weeks after treatment. By that time, the plant start to recover and resume normal growth after that. Prunes, some symptoms early on after treatment, very minor, but the plant again recover from that quickly and appear normal after 3 weeks. I think with that, I'll show you the last one, grapes, and we did see some injury symptoms, burning, not the severe symptoms you see from auxin type herbicide, and we are in a process of harvesting the crop this year and look at the yield. With that, thank you for attending my presentation, and I want to recognize the students who helped on this project, especially Denise Enns. Thank you.
Thank you, Kazim. Okay, so next is me. We'll give everyone a moment here to finish the poll. Okay. Oops. Okay, so I'm going to be talking a little bit about Wheatie rice and herbicide failures that I've been seeing this year on field visits. So the Wheatie rice field experiment in Davis is in its 3rd year this year. We established it in 2018. We've got 4 biotypes. We only had found 4 at that point. We started the actual experiment in 2019. We have a conventional flood treatment, a crop rotation treatment, and a stale seed bed. This year is our 3rd year of the experiment, still going. We had some issues last year with geese in our field, and so we restarted the rotation this year. So again, we're doing our flood rotation, starting with rice again. And then we'll be doing the stale seed bed as well. And then next year we'll be going into sorghum. The field looks considerably better this year than last year, mostly because we didn't have a goose attack this year. This is the stale seed bed treatment. I'll just remind everyone real quickly what that is. So the stale seed bed, basically we flush the field, let the Wheatie rice come up, spray it with glyphosate about a week or so after flushing the initial flush, and that should kill most of the Wheatie rice, but we're still, looking at how much it kills. And then a couple days after that, we flood up the field and then seed into the flooded field. This year, because the rotation is the same as the continuous flood, I'm just going to show the continuous flood. So continuous flood is basically just comparing sort of a traditional rice system with a continuously flooded field, comparing how the Wheatie rice emerges and how it competes with the rice in a continuously flooded field. So we've been collecting emergence data, And we'll be collecting yield data at the end of the season as well. And we'll have a little bit more information, hopefully, to share with you guys at the winter grower meetings. So for Wheatie rice, we've been looking at some potential spot spray options. This is year 3, I think, of looking at spot spray herbicides, and so far we've not had one that we were able to actually get through the registration process. But we have another new option this year. Actually, we started working with it last year, called Suppress. There's field testing done last year in one type 3 field, and I'll show you guys some pictures in a moment. The efficacy was okay last year. It didn't completely kill the rice, the Wheatie rice. The application may have been slightly too close to heading, and some of the plants actually still headed. This year, we just tested again, and we did it in one field with multiple Wheatie rice types in the field. The efficacy this year appears good, and we applied it right before heading, so a little bit earlier than we did last year, and I'll show some photos in a moment. We're also doing some greenhouse testing this year, and it should be completed soon. And we'll have the data for the winter grower meetings as well. So this was Suppress in the field last year on a type 3 Wheatie rice. This is 1 week after spraying. The plants looked very dead, but some of these heads actually still ended up producing what appeared to be viable seed. So this year we went a little bit earlier. This was 1 week after spraying, just last week actually. So we did 2 different spray timings basically to simulate a little bit more of an application in the field. So 2 different spray timings, 3 and 5 seconds with a backpack sprayer. And both timings actually appear to be doing pretty well controlling the Wheatie rice. There is, of course, a little bit of drift around the Wheatie rice plants themselves. Suppress is non-selective, so it will kill any rice, anything around it. But it looks good, and I think it's going to be hopefully a viable option. So just to clarify, if we can use Suppress right now, can we use it this year? Can we use it next year? So it is already registered to be used in rice in California. However, there is a caveat. The label does not allow for application into standing water. So at this moment in time, we still need to have some discussions about how we can use Suppress and if we can use it legally to control Wheatie rice. So we're currently doing the data collection that I just showed you, and we'll be discussing this with Westbridge and California Rice Commission to see what options we have, and hopefully we're hoping we'll be able to use it next year. So yay to maybe have a viable option here. So some herbicide issues in the field. We've been seeing a considerable number of issues this year. Surprisingly, I thought it would be a better year than it was last year. And you guys can go ahead and take that poll as we go here. Oops. Oh, I don't know if I can hit it. I can't seem to advance. Oh, there we go. Okay, so the 2 major issues that I've been seeing this year are Springletop and watergrass. So I think this is fairly consistent with what I've been seeing the past couple of years as well. So watergrass here on the left, this is one of our newer either biotypes or species, and we're collecting some more data on this, hopefully this year. And then on the right, we've got our Springletop. So some of the issues I've been seeing with Springletop failures in particular, some of the common reasons are potential herbicide resistance, of course, but then some of the other major likely issues are the water not being held on the field, sometimes water being too deep, application timing, or application method. So a variety of different things going on. So, you know, it's easy to sometimes blame the herbicide, but often there's application issues going on. This was the field I visited that I strongly suspect is not related to resistance, just because of the pattern of the sprinkle top in the field. So just some reminders for folks to remember that application and how you apply the herbicide is just as important as the herbicide that you're using.
Hi, Whitney. This is Rachel. We're sharing the poll results, and it would be good if you could address the true and false answer, since there's a little bit of
Confusion. Okay.
yeah.
Okay, so the answer to that is that there is not actually an herbicide currently registered for spot spraying as it stands. Okay, so the label is registered for use in rice, but because it's not labeled for use in flooded fields, we need to clarify a little bit further with the company and with registration, DPR and EPA, to make sure that we're following all rules. So we're going to work out some sort of either label amendment or clarification, and Roberta Fierved with the Rice Commission will be leading us on that. So currently, you still should not be using suppress for spot application in rice at this moment. Thank you. Okay, so the other big issue that I've been seeing is watergrass. So this was a field that I visited last year, and just taking a look at it, you can see the huge watergrass infestation. I mean, most of the field is watergrass. So this was the newer biotype or species, the one that we're not quite sure what it is yet. But the grower and PCA applied multiple herbicides, so multiple granular applications, multiple foliar applications, and this was what was left at the end of the season. So didn't look so great, and it was a very expensive program, obviously. This is the same field this year, and we decided to try something different, which was doing a pre-plant stale seed bed. So kind of like we're doing with the Wheatie rice. So we did a stale seed bed, or the grower and PCA did, before planting using glyphosate. And then they followed up actually just with a foliar application. So they actually didn't do a granular at the beginning of the season. But they did follow it up with a, I believe, propanol application a little bit later in the season. And the field, as you can see, is much cleaner. It looks actually pretty good compared to what it looked like the previous year. So generally just some things that I think are good to remember as we're talking about herbicides. So if you suspect herbicide resistance, please talk to either your farm advisor or PCA about seed collection at the end of the season for resistance testing. So Kasim's program at the experiment station is still taking seeds for herbicide resistance testing, and it's really important to know if you actually do have herbicide resistance, and if you don't, then what other sort of practices we need to follow to better use our herbicides. Make sure your water levels are correct for applications. So keeping the water level deep is actually super important, especially for our granular applications at the beginning of the season, and then trying to move water onto and off of the field as quickly as possible. And I realize this isn't always completely feasible depending on your water situation, but it really does make a big difference in terms of control. So just leaving the water, off or on for a little bit too long in either direction causes different weeds to germinate. So again, water and managing water is really, really crucial to weed control. And then some general suggestions for watergrass. So what I've been kind of noticing is that, and this fits in with what we know about watergrass emergence, so make as many applications early in the season as you can. So pre-plant if you can, or right at day of seeding, but basically as close as you can to the beginning of the season with as many modes of action as possible. So a lot of us have watergrass that might be, even if it's not this new species or biotype, it's maybe herbicide resistant. And so one of the ways that we can overcome that is by making sure we're applying multiple modes of action. So that could be through tank mixes, that could be through sequential applications made close together, but basically trying to hit it with as many things as possible in a short period of time. If your field is very badly infested, then a stale seed bed, like the grower and PCA did this year, that might be an option, and it does work if done correctly. And then we're looking for seed collection this year to try to get a better handle on this watergrass problem. So if you do have fields that you think have bad infestation of watergrass, we'd like to collect seed from your field. So reach out to myself or Luis, and we'll come out and help you collect. And then for springletop and flooded conditions, emergence period is very extended. So if you're applying one thing at the beginning of the season, say for example, Serrano, you're not likely to get full control for the entire season. So you, again, like one application at the beginning of the season, you're gonna have stuff that comes up later. Shallow water tends to increase emergence. Okay, so water loss also during the season can create opportunities for the plants to emerge. So one of the things I've seen about springletop is that a lot of folks that have bad infestations were having water losses or shallow water during the season. And then the last thing, stale seed bed may be effective, but we don't have data on this particular species. And my guess is that it probably is not effective, just based on what we know preliminarily about springletop emergence. So, and I think that is my last slide. All right, thank you. Okay. All right. Our next speaker is Dr. Luis Espino. Luis is the Rice Farm Advisor in Glynn and Butte counties. Go ahead, Luis.
Hello, everybody. Yeah, so I'm going to load here the presentation, and I'm hoping you can see it now. And yeah, so I'm going to talk about a couple of things. First, I'm going to give a short update on rice diseases and things that I've been seeing so far this year. And then I'll go over a couple of trials I have at the rice experiment station on tadpole shrimp and armyworms. And so Bacony has one of the things that I've seen this year that it's been a little different. Usually I see, you know, just a few plants here and there. Actually, the past few years, I've had a hard time finding Bacony affected plants. But this year I started noticing the Bacony more or less at late tillering. And you know, you can see these plants that are a little taller, they're yellow, and then you pull them out, you slice them open, and you see that the crown is rotted, and that's a sign that what you have is Bacony. And this year I've seen more than usual, and I visited a field with a heavy infection, and I'm not sure why we're seeing more Bacony. It might be you know, the issues at planting with seed being in the tracks for too long. Not clear, but there's certainly more Bacony out there than usually. And then the other one is blast, especially on the west side, on the northern part of Colusa County and in Glenn County. These are areas where we see blast most years. We started seeing leaf blast in June and July, and now we're seeing, you know, quite a bit of neck blast. I've seen a little bit of blast also on the east side, actually Afton Road area. Not much, but a little bit, and which is unusual because we don't see blast there usually. And you know, why are we seeing more blast? Most likely good conditions, warmer temperatures, like Bruce was saying, especially at night, are very good conditions for blast development. So I have some fungicide trials across the valley looking at mostly control of stem rot, aggregate sheath spot, and kernel smut. And so far I haven't seen any kernel smut. You know, fields are starting to get drained now, and this is when kernel smut start showing up. So if you see any, if you know, happen to be on a field with some heavy kernel smut, let me know. But so far I haven't seen any. And you know, this This will all be harvested, and then the results will be shared probably at the winter grower meetings. And just a reminder that we have these new fact sheets that are on our website, and there's a couple on diseases, and I think there's one on blast that's going to be available here in the next few days as well. Okay, so now I'm going to talk about the trials I have at the experiment station, and this is what you would see if we had the rice field day. So I have a trial looking at tadpole shrimp, and the question we want to answer is, when exactly is rice not susceptible to tadpole shrimp? So usually we say, you know, once the plants break through the water, those seedlings are not, you know, susceptible to shrimp injury. But that, you know, that can vary quite a bit. It depends on temperature, depends on the water depth. So we wanted to look at that more carefully. So I have a trial where we have two basins. These are half-acre basins, and on one of them we treated it, and so there's no shrimp, and the other one we left untreated, and we have a very good shrimp population there. And then on both basins we seed at different times after we start the water. So we seed at 2, 4, 6, and 8 days after we start the water. And then what you're seeing right now, so it's the treated basin, so there's no shrimp. And you can see that the plots look different sizes, and that's because they were planted at different times, but the stand looks good for all of them. But this is the untreated basin, and so there's plenty of shrimp here. You can kind of see the water is muddy. There's the shrimp were led to develop from the very beginning. And then you have the numbers there mean the days after flood when those plots were seeded. And so you can see that the only one that made a stand is the plots that were seeded 2 days after the flood was started. Everything else was obliterated by the shrimp. And so what we looked at here was every day we sampled the shrimp, we measured the shrimp, and then every 2 days we determine the stage of development of the seedlings. And that's what I'm showing in this little table here. But I want you to focus on the days after flood here. That's on the 6th day after flood. The shrimp were about 5 millimeters. Their shell was 5 millimeters, and that's about 2/10 of an inch. And then on the plots that were seeded 2 days after the water was started, the seedling stage was S3. And on that same date, the plots that were seeded 4 days after the water was started, the seedling stage was S1. But those plots or those seedlings on the untreated basin were all consumed. So we also sampled for tadpole shrimp injury. And on those plots, all the seedlings we sampled were injured. And so what are these? So if you think about it, you know, the shrimp is there. They're at a size that are causing injury on the younger plots, but not on this one, on the S3. So what are these seedling stages? So up here, what you see is the S1 and S2. So we call S1 the stage when you have the coleoptile only, which is this little one up here. It's gonna grow into the tiller. And then this is the radicle that's gonna grow into the root. If you have both structures, that's S2. If you only have the coleoptile, it's S1. But these two, S1 and S2, these two stages, the shrimp will just eat them up. They love it. And here's the S3. The S3 is when you have the profile, or what we call the spike, and it's green already. And so here's the coleoptile, and then after the coleoptile, you start getting that green structure that comes up, which we call the spike. So that's the S3 stage. And at that stage, you can see that the root is well developed, is going down into the ground, and those seedlings are well established at that point. And so though that stage seems to be the least susceptible to tadpole shrimp injury. So this is just showing a close-up of what the shrimp does. So this is a normal seed without injury. The coleoptile is coming out, and this is what the shrimp will feed on. They just clip the coleoptile. This seed might make it. It might regrow, but most of the time what happens is the shrimp will consume the whole thing. So there's nothing left. It's the coleoptile, the radicle, and this seed just sits there, it rots, it never germinates. The other thing we usually say about shrimp is that they also can uproot seedlings. So in our trial, we noticed that on the plot that were seeded 2 days after the flood was started, we didn't lose any seedlings, even though there was a lot of shrimp on the untreated basin. And so if you compare the stand on the untreated basin with all the shrimp, was about the same as the stand in the treated basin, so about 50 plants per square foot. So very good stand in both basins. So this is kind of showing that the shrimp are not uprooting those seedlings. Now, usually this is what you find in the field. In a field where there's shrimp, you'll find all these floating seedlings, and they don't have any roots. Obviously the shrimp have consumed these roots, but I think what happens is that what's happening is that the wind is uprooting a lot of these seedlings that are not well anchored, and as they float up, then the shrimp will consume the roots. And so in our treated basin, where there was no shrimp, we found plenty of seedlings that were floating. We had some pretty strong winds in the spring, but they all had the roots. So because there was no shrimp to consume the roots. So we couldn't quantify this in a way to be certain, but we're planning on doing that next year. Okay, now I'm going to jump into the armyworm trials. And so we have this trial at the station, and over there we don't get armyworms. So we have to cut the rice to mimic the defoliation. And so we did this last year, and we found that when you defoliate rice severely, all the way to the water level, 25% yield reduction. And probably could be even higher because our yield potential last year, we were in late planted rice, so the yield wasn't very good, but even at that point, we saw 25% yield loss. So this year, what we're doing is we're defoliating 3 different varieties to see if we see differences on how the varieties recover from the armyworm injury. And so we have 105, 206, and 401, and they're defoliated to 3 different levels, 25, 50, or 100%. And you can see here the plots that were defoliated, what we call 100%. We don't defoliate them all the way to the water because when the armyworm eat the rice, they always leave some rice above the water where they can hang on. So this is what it looks just after we defoliated. This is what it was looking like a couple of weeks ago before the smoke. So the 206 and the 105 were beginning to head. The 401 is just right now beginning to head. So far we haven't seen a difference in the response. For all varieties, if the defoliation is less than 50%, the plants recover fairly quickly. What you see here is the plant height up to last week. But you can see that for all of them, when the defoliation is severe, so 100%, the plants quite don't make it. They don't get to the height that they should be if they were not defoliated. And then if we look at heading, we can't tell 401 yet because we don't have the data. It's heading right now. But for 105 and 206, you can see that line, the orange line means the 100% defoliation. And the fact that it's offset to the right means that the heading is delayed. But it looks like 206, the delay is not as severe as in 105. So that's something that we're noticing on this trial. And then we have another, and so here, what we were showing here is the number of panicles per square foot. And so at the end, we all the defoliation treatments, they all result in this about the same number of panicles per square foot. But what we notice is that the panicles in the plots that are defoliated are smaller. So what we're gonna do is when we come to close to harvest, we're gonna take panicle samples and we're gonna count the number of kernels per panicle to determine, the effect on yield. And then we're also gonna collect yields, so we'll know the yields for each plot. And then I have another defoliation trial, but in this one, we're looking at the effect of a top dress after the defoliation, with the thought of, if you get an outbreak, the armyworm produces some severe defoliation, putting a top dress, does that help? Is that gonna make up for some of the yield loss? Is it gonna help the plants recover? And so we have plots here that we defoliated to 2 levels, 50% or 100%. And then we have half the plots have a top dress, 30 pounds of nitrogen, and then the other half don't have the top dress. And this basin has all the nitrogen, 150 units, in the soil, pre-plant. And then, okay, so what I'm showing here is the plant height. So we're looking at plant height on these plots. And so far, we don't see really a difference, a benefit of the top dress. The top dress is not making the plants recover any faster or any better. This is plant height to last week. But then on heading, we see again on the not top dress, this is number of panicles per square foot. We see that there is a delay on the plots that we defoliated 100%, but the delay is not as severe when we put the top dress. So that's an interesting observation. And again, we're seeing that, the plots where we defoliated, the panicles are shorter. So we'll take those samples at the end and evaluate and see if the top dress helps with that. And then we'll check the yields as well. And so that's what I wanted to share today. I see there are some of the questions are up, and do I have time to go over that?
I think we're a little bit over, Luis, but
Okay, that's fine.
yeah.
Let's
But
see.
I think we can answer more at the end.
I think most people got him. Yeah, we'll go back to this at the end.
Okay, thank you. Okay, our next speaker is Dr. Ian Gretenberger. Ian will be covering, Ian is an extension specialist with UC Davis. In rice, he works on management of key arthropod pests, including rice water weevil, tadpole shrimp, and armyworms. His talk today is going to be on armyworm trapping, rice water weevil trials, and tadpole shrimp insecticides. Ian, whenever you're ready.
Is that good?
We do not see your screen
All right,
yet.
your screen. Thanks. Sorry. Oh, great. Yeah, that doesn't work. Let me see. Sorry, I'd switch computers because my, okay. Good?
Yeah, we see it now. It just needs in presenter view. Yep, perfect. We see it.
Thank you. Sorry about that. I using a different laptop. My other one wanted to update, and I figured that could be a disaster. Okay, so yeah, thanks. I do want to thank the Rice Research Board for funding this work, and then also want to acknowledge that a lot of this work's in cooperation with Luis Espino too. So yeah, just first I want to touch briefly on some of the stuff we're doing on rice water weevil, and some of this stuff's going to be kind of a big overview at this point, just in terms of the amount of work that we're doing, and then there will be updates on the results later, the winter grower meetings and later in the year. but here's the rice water weevil. It's one of those pests that's historically been a big pest. It's less of an issue now. We're really not sure exactly what the reasons are, but we've been continuing to monitor population trends, and the last probably 8+ years, the populations have been pretty low, but it is still a pest. This is from last year, but there's the damage, and so it is a pest that we have to worry about. And then the other key thing with rice water weevil, it obviously causes that damage as larva, but then also it's just the for registrations, it's a pest that's relevant across the U.S., across the southern growing region, where that's their primary pest. And so it's really critical for evaluating new insecticides and getting them into California. So we're continuing that work. And so for this work, we're using this is what right outside of the field 8 there, right next to the building at the Rice Research Station. But so, we use these rings and infest them with rice water weevils. And so most of this work is focused on insecticides at this point, plus a little bit of testing of susceptibility of different varieties, which I won't go into that at the moment, but that's also, important to keep testing new varieties as they come out to make sure that we don't have surprises with weevil damage. And so I just want to show what we're testing this year. We kind of limited some of the insecticides. Right now, there's not a lot of new insecticides for rice water weevil. The South has shifted in large part to seed treatments for rice water weevil, which isn't really something that we're using here in California. But as you can see, some of the standards that are used a lot in rice, so Warrior or Lambda cyhalothrin, testing that. And then also diflubenzuron, which is a growth regulator, and then also testing an organic treatment there, Venerate. And then we're probably going to do some more greenhouse tests with some other biologicals as well, just to see how those affect rice water weevil. And so then moving on, so one of the other pests that obviously Luis has touched on that we're also working on is armyworm. It's one of those key pests in rice right now and can be a little tricky. So, this is the way you see initially when armyworms are in there. You see a little bit of nibbling, but obviously the worry is that nibbling will turn into full-scale defoliation. And so figuring out where and when these populations occur is one of the key things and something that we're trying to get a better handle on because detection is so key. And so, so I mean, when you're the egg masses are virtually impossible to find. We don't see them. And so this is what's happening initially in the fields. We find these little tiny larvae, but these guys are really hard to find. And so ideally there's a better solution than poking through the rice and really looking for these little tiny larvae. And these little tiny larvae, they're not doing much damage, but the worry is, right, if they keep growing, they turn into these big guys, and these are the ones that can defoliate rice pretty darn quickly. So we want a better sense of, when to know when these big ones might show up, the damaging populations. And so some of this is monitoring. So, for here, we're using the pheromone traps. So it's a female produced pheromone. We're trapping the male moths, so the male moths are attracted. And that's the gist of this tool in terms of monitoring. And so Luis has been running a monitoring network. If you haven't gotten queued in on this, definitely go to the Rice webpage there and sign up for the newsletter. But he runs a trapping network. And so that's partly what's been inspiring some of this various monitoring work that we're doing right now. And so one of the questions is, if you have traps, you're putting traps out, how many traps do you need? How well do those traps capture the spatial variation across the landscape, right? Because this is kind of what you might look at. Some people might be growing across a lot of acres, or fields might be spread out. So we want to know, if you get traps, put out traps, how many do you need to put out, and so forth. And so one thing that we're doing this year, the work that Luis is doing has a couple traps in a given location spread out across the whole rice growing area. Here what we're doing is we're putting out a lot of traps in a relatively small area to get a sense of the variation between traps. And so we have this grid on a half-mile grid that we have out there for monitoring for true armyworm. And so this just give you a sense of what we're seeing here. So these numbers are moving. So that just shifted through a couple of days, but the gist of it there is, you know, we're actually seeing some hot spots like that top left one in red there, and figuring out, you know, there's nothing clearly different about those spots, but there is some variation, which is going to be important when we're thinking about, you know, if we do want to use these traps for monitoring and predicting when armyworms are going to show up. And so as part of this work, we're also doing, there's a continuation of work that Luis started, is within this grid and associated with some other traps too, we're also looking for larvae close to these traps to figure out, you know, can we link trap captures to larval counts? And if not, then the next step would be figuring out why is that and what's driving those larval numbers. And another thing that we're still working on, this is continuation of work from last year. So we are working with some automated traps. That's it there on the left, and on the right is it's opened up to show there's a sticky surface there that can be circulated, or it's got rollers. And then there's a camera on the top that takes a photo. So these data are reported remotely. And the idea here is this is kind of real-time data. And so if we can better get a handle on the moth peaks and moth flights, then that might be better able to predict when egg laying is occurring, and these might play a role. And here's what the image looks like, and they do have some AI in there to recognize the moths. And so we are working with these again this year. And then the other pest that we also have here are the tadpole shrimp. These guys are obviously kind of funny as far as pests goes, which obviously Luis gave a bit of the background on how they work as pests. And they can come in quick and hard at the beginning of the season, which makes them a little bit different to control compared to some other pests. But really it's, you know, as soon as you add water, there's the possibility for infestations if you have them in your field. So one of the key things that we're doing this year is doing some insecticide testing. In the past, Luis has used some berm studies where he was giving the berms, the populations there, a chance to build this year. And so we did a lot of the insecticide work in these rings, the same rings that we were using for rice water weevil. And so we actually tried a couple different methods this year. And so on the left there, we have some rings, and so we're relying on natural infestations of tadpole shrimp there. The key indicator that there's natural infestations is just how murky that water is. So the shrimp on the outside of the rings, we're really stirring it up there. A little bit of wind, but that's really all shrimp. And so, you know, we're testing different insecticides within those rings. With natural infestations. And then we also, on the right there, there's some rings that we set up in a field that doesn't really have many tadpole shrimp. And so there we were moving shrimp into the rings. And then we also had some other tests with those buckets there where we cut out the bottoms and put some holes in them and put mesh in them so that we could actually get better counts on them. But so we were putting these buckets in rings and then treating the rings with insecticides. So for the earlier ring study, which is early timing, and this is where we're relying on natural infestation. We're gathering data on shrimp plus stand counts. And so this is just the numbers or the various materials that we're testing. There's the active ingredients. There's also the timing, because shrimp are most susceptible when they're really tiny, but obviously getting the application on quickly sometimes can be a challenge, or sometimes shrimp can surprise you because, as Luis showed you, they can take off really quickly and grow very quickly. And just to note a couple things there. So diflubenzuron is a growth regulator. So the timing is probably is important. And so that we're testing that, also testing some various rates because it's not cheap. So that comes into play. And we're also testing a few new insect growth regulators. So Novalaurinum, buprofezin are growth regulators. And then that cocoa bear, that's actually an oil that's used for mosquito control. And what it does is gum up the surface. And so when mosquitoes go to the surface to breathe, it gums up their breathing apparatus. And if you've seen tadpole shrimp, oftentimes, especially once they're bigger, they'll go gill side, or so upside down on the water surface. And so the idea here is we might be able to gum them up with something like this. And this is a conventional material there. And then down at the bottom, we're also looking at biological control. So it's very early, getting with, there's mosquito fish in there very, very early. And this is for those rings. The density is for those rings. And in the later study, we had timings later. So when the shrimp were a little bit bigger, means that they're probably going to be a little bit harder to control, especially with the growth regulators that we have there. And I will note, so buprofezin, the growth regulator there, we actually jumped the rate because we saw in the first, with the lower rate from the first study, it didn't work. Looked like the untreated, so we tried a higher rate. And then we also tried an MSO as an oil, just to see what that would do to the shrimp there. And results we'll share the results to fall, but we'll say that Novaluron, the growth regulator, was highly effective. It's actually a known characteristic in that it's highly toxic to aquatic invertebrates, which normally is a problem, but if that can be taken advantage of in rice for shrimp management, that'd obviously be a good thing. And so just a photo and then just a moving image there on the left. They're kind of floating around. So this was actually with that cocoa bear oil. So they actually kind of got gummed up and then stayed at the surface. I think they were having trouble breathing at that point. So we saw some variable effects. It didn't seem to work as well as one might hope in terms of 100% control, but we were seeing some effects on the shrimp. And then this is what we see. We're looking at some plant stand. We're looking at those floating seedlings. And on the right, that's a plot that had really good shrimp control. Rice looked great. On the left, you see that small plants and really shoddy stand. And then finally, just want to touch briefly on something we're also continuing to do with shrimp, just looking at insecticide resistance, because lambda-cyhalothrin is used a lot in rice, as you can see here, and it's really effective for shrimp for the most part. It's very cheap, but it's used a lot, and so that puts a lot of pressure for resistance possibly. And so we are continuing work monitoring for resistance. So this is Luis out there gathering some soil. This was before rice was planted. So we're collecting a bunch of populations of tadpole shrimp and then seeing how resistant they are to pyrethroids, specifically lambda-cyhalothrin. And this is something, if anybody has had issues with shrimp management this year with pyrethroids, definitely let me know, and we can come take samples and try to test them. And so finally, just want to acknowledge the Rice Research Board for funding, grower and PCA cooperators for this work. Kevin Godding's my research associate that leads this, and then some undergrads, and Luis too. And I have this, so I'll just put it up, and I see that the poll's up as well.
Okay, thank you, Ian. All right, and we've got one more presenter, Dr. Michelle Leinfelder-Miles. She'll be presenting on her Delta Rice update. She is a Cooperative Extension Delta Farm Advisor based in San Joaquin County, and her research and extension program focuses on field crops and soil and water resource management. All right, Michelle, whenever you're ready.
Okay, am I sharing? How do I look? So I'm the farm advisor. Thanks, Whitney. Michelle Leinfelder-Miles. I haven't presented at one of the rice grower meetings up in the Sacramento Valley during the wintertime, so I'm glad to have this opportunity to present now. And I'm going to be talking about some of the activities that I have going on in the Delta region. So first, a little bit of overview on Delta rice. Acreage is a fraction of what acreage is in the Sacramento Valley. This table is showing San Joaquin County rice production. Most, if not all, of the rice production in San Joaquin County is now in the Delta region. There used to be an area in the southeast corner of the county where there was some rice production previously, but that almost has disappeared and turned into tree crops. So now most of this acreage, I would say, is in the Delta region. There's also some acreage in that tail of Sacramento County that's in the Delta. Acreage is increasing little by little. Rice production is being supported by the Delta Conservancy for reasons of mitigating soil subsidence and carbon emissions. Some of the main differences in the management of rice in the Delta region compared to the Sacramento Valley includes drill seeding. So all of the Delta rice is drill seeded, and the permanent flood is established at about 3 to 4 leaves. Rice in past years has been grown as part of a rotation on some of the farms in the Delta, maybe 2 years of rice, followed by 2 years of tomatoes, and then back to rice again. As I've spoken with growers this year, rice production tends to go back to back. A lot of growers have gotten out of the tomato business, and so these rice growers are now growing their rice back to back, and rotation is not as common. Challenges in the Delta include cool summer nights that can cause blanking. It also limits our variety options. Windy conditions can compromise our timing of pesticide applications, and then pest problems. We probably have fewer pests than what growers in the Sacramento Valley struggle with, but certainly we have our share of pests that can be challenging. These are the activities that I'm going to talk about today. I'm going to start talking about a herbicide trial, and then I'll briefly touch on the armyworm monitoring that I'm doing and some Wheatie rice outreach that I'm doing here in the Delta. So obviously weeds are important pests in California rice systems, and herbicide resistance can occur, as Whitney and Kasim have spoken about already. And then Kasim talked a lot about this chemistry called Loyant, this new product that's registered in the southern United States, but it would be a new chemistry in California. So experience in the southern US has shown efficacy on some of the broadleaf weeds and the sedges and rushes, and some activity on the grass species, barnyard grass and watergrass. The question for this trial specifically was, what is the efficacy of Loyant in the drill seeded systems that we have in the Delta region? We had a few different treatments that we looked at. We looked at different rates in this trial in 2019. We compared Loyant to the grower standard practice, which is the Regiment, Sandia, Prowl, and Super Wham treatment, that fourth line. We didn't have an untreated control. Instead, we called Prowl, our pre-emergence treatment, our control treatment, and compared to that. And then we had one treatment there listed at the bottom where we substituted the Loyant into what would have been the grower standard treatment, except we used Loyant instead of propanil. In this trial, these were the weeds that we had particular problems with, the grasses. Sprangletop was a particular problem in 2019. And then there are a few sedges in this field, although certainly the grasses are the most problematic weeds that we were looking at in this trial. Crop injury, we looked at crop injury starting at 7 days after treatment, and then in subsequent weeks after that, 7-day intervals. The Loyant treatment did very well, no crop chlorosis or necrosis. However, we did notice some leaf curling at about 14 days after treatment. That symptom, however, was gone by 21 days. So by the 3rd week, we weren't noticing that leaf curling at all. The grower standard treatment, we did notice some pretty noticeable effect of crop chlorosis and some tip burning. And that effect was a little bit less when we substituted the Loyant for the propanil. We didn't, however, see any severe crop injury in this trial. There was no stunting or stand reduction, and no differences among these treatments in heading. This table is probably a little bit busy, but the point to be made here is that we looked at weeds, we counted weeds in each plot over 7-day intervals. And really the only significant differences statistically were that all the treatments did better than the Prowl, what we called our control treatment. And then there are a couple columns there where I excluded Sprangletop. So Loyant, as Kasim has already talked about, does not have activity on Sprangletop, and Sprangletop was a particular problem in this field. So I excluded Sprangletop from the counts just to kind of get an idea for how the other weeds were being affected and how the treatments were doing when you excluded a weed that we knew the herbicide wouldn't have any activity on. Our yields, there were no differences in our seed moisture and our yields at the end of the season. And our yields were basically quite good. The Loyant treatments had yields that trended better than the grower standard herbicide practice. And all of them were better than county averages. So to conclude our results from the 2019 trial, we did observe a little bit of leaf rolling from the Loyant treatments, but those symptoms were gone about a week later by the 3rd week after treatment. Loyant had good activity on the grasses, and the activity the control was similar to the grower standard practice, but keeping in mind that tank mixes would be necessary in order to manage the springle top. So we could see Loyant as fitting into a herbicide program for growers here in the Delta where we have our drill seeded systems. I'm just going to touch very briefly on the trial that we're conducting this year in 2020. We're looking at Loyant in combination with other herbicides this year, trying to get a handle or a feel for how these combinations would help control the weeds that we're seeing in the trial. Interestingly enough, we're not seeing sprangle top the way that we saw it last year. And then another thing that we did observe is that the Loyant had some of this leaf rolling that we observed in 2019, but it was a little bit more severe in 2020. We don't have a really good handle on why that is, but likely some environmental factors that caused that leaf rolling to be prolonged. But after a few weeks, it was probably a few weeks that we saw that leaf rolling compared to just 1 week in 2019. But after a few weeks, the rice has come out of it and there seems to be no differences in heading among those treatments. And then finally, the last thing that I'll say, and this would be a little bit in contradiction to what Kasim has mentioned earlier on, but at least in this drill seeded trial, I've observed that Loyant and Super Wham, which is that treatment that's at the bottom of the table, is actually doing quite well and controlling the grasses better than some of the other treatments that are listed higher up in that table. So we'll see how this all pans out at the end of the season when we get our final results. So Armyworm monitoring, we've been monitoring in the Delta in about 3 different fields since 2016. And then I collaborate, Luis takes the lead on this. I send our results up to Luis and he reports out the Delta data with his weekly updates. So if there's anyone out there who is growing or consulting in the Delta region, I recommend that you get subscribed to Luis's updates. And then we've partnered or collaborated with the California Rice Commission to ensure that the Section 18 on Intrepid has also been approved for San Joaquin County and Delta rice. This is a graph that shows the true armyworm counts that we've seen over the last few years. Our highest peaks have been in June and early July, just as Luis has indicated in his results. And this year we didn't see numbers quite as high as what we've seen in previous years. I will say, however, that, so before I move on, I would say 2 out of the 3 sites where I'm doing the monitoring treated for armyworm this year. In the one site where the grower didn't treat for armyworm, we do see a little bit of panicle injury. That picture at the bottom left enhances what the damage looks like when the armyworms are causing injury to the panicle. But just walking the fields, I have noticed a little bit of panicle injury where the grower wasn't treating, but otherwise damage seems to be very slight this year, if any. And then this is a picture actually of a cover crop trial that I have immediately adjacent to some rice. I hope my mouse is showing here, but this is the rice in the background, and this was a cover crop trial that we had. It's a mix of cowpea and triticale, and we were in there a few weeks ago to do sampling of the cover crop. And before we even walked into the plots, we were noticing a lot of injury to the triticale, the grasses here. The cowpea seems to be just fine. The triticale was severely impacted by feeding, and you get down low, you cut that biomass away, and you can see the armyworm pressure that was in this field adjacent. And I bring this up only because in the Delta region, but probably in the Sacramento Valley too, there's a lot of riparian areas, there's some wildlife habitat, and there could be you know, areas very close to our rice systems that will harbor the pests that can do some damage to our rice. Now here we didn't see damage in those particular rice fields, and you know, it could be that triticale is a preferred host for the armyworms, I don't know. But certainly we saw a lot of damage in this field right next door. So finally, Wheatie rice. Whitney's been a huge help in helping doing some outreach on Wheatie rice here in the Delta region. So just to kind of get down to the basics. So Wheatie rice is Wheatie because it has undesirable characteristics. And one of those characteristics is that it shatters very easily. Sometimes it can be called red rice because it may have a red pericarp, but certainly it's that shattering and the seed dormancy that make it really undesirable and weedy. So we've observed Wheatie rice in the Delta since 2016. And my primary activities include assessing the distribution of Wheatie rice and disseminating information on what we know to be the best management practices. Whitney's talked about different biotypes, how they have different characteristics. What we have found in the Delta is type 1 Wheatie rice, and characteristics of that biotype are tall stature. It grows taller than the cultivated rice, and sometimes it's a little bit yellower too. So even if you're not observing that it's taller quite yet, you may notice that the color is a little bit lighter. It is highly shattering and has very high dormancy. So if those seeds fall off the panicle, they will get into the soil and contribute to a weed seed bank. And those weeds can last for a very long time. We're aware of a few ranches in the Delta where there is Wheatie rice with infestations that range from low to quite severe. Our best management practices, and you know, we're learning as we go, as this is a relatively new pest, well, not new, but a re-emerged pest, let's say, in rice systems. So use only certified seed. In fact, this is the law now. Since type 1 shatters very easily, rogue those plants as early as you see them, if possible. And after heading, try to bag the panicles and remove the panicles, if the infestation is not severe. Make sure your equipment is clean and harvest Wheatie rice infested fields last, if possible. Crop rotation or fallowing may be needed if infestations are severe. And since type 1 has high dormancy, avoid tillage or use only light or shallow tillage whenever possible. In the Delta, we have observed in one of the first fields where we observed Wheatie rice, we did find that the weed seed bank was reduced when the grower had post-management that included mowing, but no tillage in the fall, followed by winter flooding. And that winter flooding seems to help with seed deterioration and reducing the infestation in the following year. So with that, I thank you, and I see there's a poll. So go ahead and take your poll.
Thank you, Michelle. All right, everyone. So we've got about 5 more minutes, and we'll take that time to answer some of the questions. So if all of the speakers could unmute themselves, we can go through the questions one at a time here. So I'll start with, I believe this is for Kasim. Kasim, the question is, for drift trials, what was the rate of methylated seed oil used and how was this decided?
It's a 1 pint per acre and decided based on the label for how it's gonna be the concentration on the label online at the label.
Okay, thank you. Let's see. Do growers have adequate insecticide options for rapid armyworm control in California?
Yeah, I can answer that. So currently we have methoxyphenicide, which is Intrepid. It's got a Section 18. It doesn't have a full Section 3 registration, but we're hoping that we get that in a couple of years. And the industry is working on another insecticide, the active ingredient Encouragin. That is also a good insecticide for armyworm. It controls good and quickly, but that one's still in the pipeline. So we're relying on a Section 18 right now, and we're hoping that we get a full registration next year or maybe the year after that.
Okay, thank you. One more armyworm question here. Did other growers treat for armyworms other than the growers you were monitoring for?
I think that's for Michelle. She mentioned she had.
Oh, was that from? Oh, okay.
I think so. Yeah, Michelle, you mentioned that you had on the couple of fields you have traps. They treated, and I think they're asking if other growers also treated.
Yeah, you know, I can't speak for all of the growers. I can basically only speak for the growers where I've done the trapping this year. I know in previous years, I've spoken with growers, maybe they would treat along the field edges as a way to reduce how much acreage they're treating, if at all. I think the growers in this region have found that even in those years where the graph was showing those high populations or higher populations than what we had this year, that, you know, maybe it's the level of risk that the growers willing to have. Some of them are maybe more likely to spray. Others are less likely to spray if their experience tells them that, you know, the damage that they're viewing or the populations that we're reporting are within their risk tolerance.
Yeah, and I'll make a comment that here in the Sacramento Valley, we didn't have a very heavy armyworm year. It's been low, but there's been some fields that were treated, but not much. I don't know the exact acreage, but I know it hasn't been as much as previous years.
Okay, and then we've got a few questions on Wheatie rice. They're kind of similar. So can farmers identify Wheatie rice easily enough to be able to rogue it before it heads? And sort of a similar question for spot spraying. Is Wheatie rice easily identified, or are there good tools or techniques to recognize it in the field before heading? So I'll just briefly say it looks a lot like watergrass. So we basically have asked folks to wait until after all of their herbicide applications for watergrass are completed, and then you basically have to go out into the field to look specifically at the plants. From afar, it's pretty impossible to tell if they're Wheatie rice or watergrass. When you get out there, you look for down near the base of the leaf where it connects to the stalk. You look for a little structure called a ligule and an auricle, and if it has a ligule and an auricle, then it's rice. If it doesn't, then it's watergrass. And then also feeling the leaves of the Wheatie rice are pubescent, so that's another way, but it is a bit difficult to identify. Anyone else want to add anything?
One of the slides that I showed Whitney at the end of my presentation showed some light greenish areas, and that is usually an indication that you should get out there and at least and check, as Whitney said, check for those structures. on the rice or on the plants. But if you see something like that, maybe it's an indication to take a little walk.
Yes. And then we've got a question about solar radiation. So I think, Bruce, probably this is for you. At what stage of development is reduced solar radiation most damaging to yields?
Boy, I would have to guess probably between PI and heading. That's the stage where the plant is growing by far the fastest, is putting on the most biomass. So I would think that would be probably the key stage. I mean, all areas, all times can be affected by it. We have a lot of solar radiation here in California, maybe even excess of what sometimes a plant may need. So a slight dip may not be a huge issue, but yeah, I would guess PI to heading.
Okay, thank you. Well, we won't keep everyone. It's 3 o'clock, but if people would like to stay around, I think the speakers are okay with hanging on for a few moments if there are additional questions, but we'll go ahead and dismiss everyone that needs to go. So thank you all very much for attending. We all appreciate your attention and your questions, and we hope to see you in person next year.
Yeah, and while we wait for, if there are any more questions, I just wanted to add on the poll, you know, I didn't have time to go through the questions, but looks like that everybody got the, yeah, there's my poll. So on question number 1, so what disease does sodium hypochlorite, the bleach, soak target? And that targets Bacony. It has no effect on smut or stem rot that we know of. We're doing some trials with smut, but we don't think it does. And blast, it could have an effect on blast if we increase the concentration, but if we do that, then we start getting a drop in germination. So at the concentration and the amount of time we use the soaks, we don't see an effect on blast. So the answer for that would be only Bacony.
Thanks, Luis. And we've got another question actually in the chat here. So is there any value in using a drone when scouting for armyworm? Anyone, go for it.
I would say, yeah, because you know, when you're scouting for armyworm right now, what you're doing is mostly looking for damage, for defoliation. And later when the rice is heading, you're looking for, you know, the panicle blanking. And so it's easy to see that when it's near the borders, if you're driving around the field, but a drone might be useful to look a little further, look closer. So I think it could have some value.
No, I think the challenge is it's the small scale defoliation, which is that first sign or even medium scale is pretty challenging to see. But I think if you're able to detect that with a drone, then I think it would be helpful. And we do have the benefit. Most of the time there's at least moderate edge effect with armyworm, so that does help, but there's still checks going out that would be challenging to drive on.
Okay, I don't think we have any more questions. If anyone would like to address any of their poll results, we can do that quickly. Otherwise, we can end this. Anyone else feel like they need to readdress some of the polls?
I think for mine, the traps are for male moths only. Yeah, it's a female produced pheromone and it tracks the males. So that's the one thing there. So that means that when we're monitoring, we are catching the male moths. So we're not catching females, which are what make the worms, but we're still relating those.
Okay.
Yeah,
Thanks,
Whitney, yeah, if you put up mine, I forget what the other questions were, so I can go through it quickly. Yeah, that's Ian's. Yeah, here we go. So my second question was, what seedling stage is the most susceptible to shrimp injury? And the trial we've had this year and some other trials have had previous years seem to indicate is when you have the coleoptile and the radical. And once that seedling has the spike and it's green, and that the root is well into the soil, is anchored, the shrimp don't seem to be a problem. And then the third question I had was, what are the effects of severe armyworm defoliation and, you know, shorter plants, delayed heading, and smaller panicles, all of the above. That's what we're seeing.
Okay, thank you. Anyone else?
I'll address.
Oh, we've got,
mine.
oh, go ahead.
I think most people got the answer, but Loyant doesn't have efficacy on Sprangletop, so it was all of the above except. And so Sprangletop was the correct answer. And that second one, best management practices for Wheatie rice, include all of the following except deep tillage was the correct answer. That would not be a good way to manage for Wheatie rice.
Okay, and we have one more question in the chat. It says, remind us again how many DPR hours were approved. So we had one DPR hour and 1 hour for the CCA licenses, and that was in the pest management category. Okay, we've still got a few folks here.
One's better than none
True.
this year.
Okay. Well, we'll say goodbye. I think that was mostly the end of the questions, it looks like. So thank you again. Hope to see you next year in person.